Hello, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia and our sponsor, Precision for Medicine, I'd like to welcome you to Biobanking in a CDX World. Are you parting the seas or aligning the stars? A Precision for Medicine point of view. At this time, I would like to introduce our presenters for today's Web Symposium. Pia Marisa Gargiulo joined Precision Medicine in the fall of 2012 as the Managing Director of Companion Diagnostic Solutions. Before joining P4M, Ms. Gargiulo led the Companion Diagnostic Partnership team at Qiagen as the Vice President. There she worked with pharmaceutical companies to establish drug diagnostic collaborations and co-development programs, consulting on the novel technology, regulatory, clinical, and commercial requirements for bringing a companion diagnostic to market. Prior to Qiagen, Ms. Gargiulo was a therapeutic brand leader and senior regional scientific manager in the Medical Affairs Oncology Department of AstraZeneca LP, working with breast, lung, colorectal, and prostate cancer pipelines. She has over 15 years' experience in industry and in academia with a strong focus in oncology. Our second presenter today is Kathi Shea, Vice President of Bioservices for Precision for Medicine. For over 20 years, she has led repository programs, helping clients build and manage biological collections from design through implementation, maintenance, and closeout stages. She is the immediate past president of the International Society of Biological and Environmental Repositories, is a member of the College of American Pathologists Biorepository Accreditation Committee, and the Biorepository Director for the CAP Biorepository Accreditation held by Precision. With that, Pia, I'll turn the presenter ball and the program over to you.
Thanks, Rachel. Thanks, everyone, for joining, and welcome to the webinar. So today the agenda is introduction on precision, some highlighted case studies that drive home the reason that we're on the line in biobanking for a CDX world, and sort of the ability to ask questions and understand a little bit deeper both our thinking and our experience as it relates to biobanking in a CDX world. So thanks again for coming. Precision for Medicine, we are a very unique and new services company focused on developmental and commercial expertise in the area of precision medicine. Our mission in this space is to be able to provide end-to-end scientific and services that fit the needs of those developing products in a precision medicine world. So when we think about and we hear probably omnipresent, whether we're in industry, government, academia, we hear the term that healthcare is changing or the healthcare industry is undergoing a combination of enormous changes which are altering both the development, the practice, and the economics of medicine. So when you look at the scientific and technological advances that sort of allow us to use large data sets, or what people are saying, big data sets, big data, moving into the omics to sort of take this large-scale approach and focus it down to a target, a patient that we can derive a clinical benefit in the clinic. So very big to very small. The industry, as it relates to just healthcare, is very focused and moving in the direction of a targeted and outcome approach. We see this in the regulatory path, in the development path, and the payer path. Precision, our team, our talent, our expertise, is built to help serve this new dynamic, to usher in the services and the support needed to see the realization of precision medicine. So our DNA, a little bit about sort of our core. As a geneticist, I use the term DNA as it relates to precision and what we're grounded in. And so with that said, our sort of core areas of expertise center around business, product, and strategy, bioservices, and we'll hear from Kathi today on that, regulatory science, market access, and companion diagnostic support. we're driven by the industry needs to focus on both the development and the commercialization, and we deliver high-value solutions that help accelerate research, reduce risk, establish a communication pathway that's focused on value, and ensuring the adoption, access, and uptake of products in a precision world. The unique combination of not just the skill sets, but it's the crosstalk between our core business areas that provide that high-value solution. So just a quick overview of sort of our operational scale and our footprint across precision today. So today there are over 120 staff members in 3 facilities. We have over 30 scientific strategy consultants. We offer a best-in-class facility and technology and manage over 15 million samples today. We have global capabilities and also focus in key global markets. We're very well capitalized, and we're continually adding capabilities to meet the growth needs of the industry and our clients. So when Kathi and I first came together, my background being in the companion diagnostic space and Kathi's expertise in biobanking, and when we first sort of met, we said, well, both of us are sort of polar opposites, me more proximal to the patient, to the industry side. Kathi on the earlier side saying, Pia, watch what you do, what you put away, how you preserve, how you conserve is critically important, because one day you might have a problem and you won't be able to do the thing you want to do with your newfangled technology. So why are you participating alongside us at Precision today? And whether biobanking is important to the aspects of your developmental process, you're thinking about diagnostics, you're thinking about a precision strategy in your organization, or biobanking is indeed your primary remit, it's your central role, and you're looking to get guidance and support about this new world of precision medicine as it relates to your bank and your source. Or the sort of other bucket, which is biobanking is becoming an increasingly important to your organization, whether you be, again, industry, government, academia. We see these factors becoming critically important to organizations, whether they be DX, RX, discovery, early targets. There's not only considerations of just the development and commercialization, there's also economic environments as to why we're sort of bringing this value point together. In a CDX world, we say tissue is the issue. In a biobanking world, I hear Kathi say specimen is the source. So it sounds like the same thing. We're all targeting valuable information to bring that information to light through the technology, to bring it down to a patient level. So it relies on asking a very simple question, and that centers around the sample. So with that said, we'll sort of move now into the case studies to help highlight some of the considerations at play in the biobanking world. So Tarceva was a very well-anticipated entree into the targeted therapy marketplace from a drug perspective. We were moving away from chemotherapeutics and saying this is the new world of targeted therapy. The mechanism of action of Tarceva was centered around epidermal growth factor receptor. Its also mechanism of action was a TKI, a tyrosine kinase inhibitor, and that ushered in this whole philosophy of target drug, being able to risk stratify patients. In this particular instance, it's a mutation test, also called EGFR, but risk stratifying patients who will benefit, who will respond. The issues and why we bring biospecimen-related issues to the development of CDX are critically important here to our moving forward and driving value in this space as it relates to both biospecimen, biobanking, and the uptake of companion diagnostics. So this instance, we sort of see this samples were collected in only half of the patient population. So someone like myself who focuses really wonders the value of the clinical trial endpoints and what that means. So these issues related to not only decreased collection, certainly implied the questioning of the robustness of the clinical trial endpoints. Also, with only 50% of the tissues sampled from areas that weren't essentially defined as primary biopsy or secondary biopsy, one would imagine that these endpoints are not only misconstrued and really question retrospectively the lack of standards in this space and time. So we present this as sort of an example of personalized medicine, a movement towards targeted therapy, but retrospectively to say there were a lack of standards in place that probably won't occur in the future, and furthermore, where we access the tissue from, right, critically important. If you're thinking of melanoma, right, you want that primary tissue. The secondary biopsy is not going to help you, and indeed you have to store it under a standardized way. So Tarceva serves as a great example of biospecimen issues related to companion diagnostics, specifically on tissue sampling. The next example, so Herceptin has not only saved millions of lives, but serves clearly as a poster child for precision medicine. Herceptin, the target was human epidermal growth factor receptor. The HER2, as it's well known, the diagnostic test initially was an IHC test. So those of you on the phone that deal with technology day in, day out, IHC targets the protein product of the gene and is not without, it's a wonderful tool in the laboratory, but indeed can cause problems technically. One of the issues when Herceptin was launched and the choice of an immunohistochemistry assay is that the results by using an IHC on an archived tissue sample that we aren't quite sure what the standard of accessing and storing the sample was in 1998. Nonetheless, the technology applied yielded a false positive rate of 38% and a false negative rate of 32%. So if you take that data set and put it back on sort of the biospecimen related issues and ask yourself, how were they stored? When were they stored? When they were stored, were they consented the right way? Was this a lesson to be learned for us on the standard of consent, how we preserve to conserve? And again, when you think about precision medicine, when you think about storing a bank, and you flank that on the world of companion diagnostics, note, companion diagnostics 99% of the time focuses on a subset of patients that will respond. It is that subset that we don't know who they are when we store and prepare our banks. So I bring this forth as another biospecimen-related issue, and it falls into both the companion diagnostic bucket, the question of access, the question of consent, and more importantly, with Kathi will also talk about, is how we preserve our specimens to conserve our specimens for the future. So I'm going to hand it off to you, Kathi.
Thank you, Pia. The case study that I wanted to bring up is one that I think everybody is very familiar with because it's a very recent event for us, and that is Hurricane Sandy and the devastating effects that it had on the east coast of the U.S. and in the Caribbean Sea as well. This storm caused billions of dollars worth of damage in the New York area alone, and there were multiple reports and news stories about the devastating effects that it had on NYU's research facilities in terms of the loss of research. And that loss of research was really as a result of loss of samples, loss of animals as well that were part of their research programs. But these losses were all due to multiple natural disaster implications on an area and a facility. So NYU was well equipped with backup generators up on the roof of their facility. However, the fuel access was on the ground and the fuel pump needed to get the fuel up to those generators so that they could actually run were underwater and therefore would not perform. So while they had primary backup systems in place for the power outages, they did not anticipate or prepare for simultaneously having a flood and how that would impact their primary backup system. Another issue that everyone faced in this area, because it's had a very widespread impact on the East Coast, but another issue that people encounter was that there were scarce resources. Trying to get refrigerant that they could use to pack freezers was near impossible because nobody had any in the area that they could get quickly to the sites. And while there were dry ice vendors out on the street, you couldn't do any electronic transactions. And, talking to some of our colleagues, they were actually in the situation where everybody's scrounging up what cash they have to try and get some sufficient resources to save at least part of their collections. And so these are the type of what-if situations that, if properly planned for, perhaps could have ended with different results. And while NYU was very vocal about the impact on their research, we know from other colleagues that this affected all types of research organizations, pharma, IBD, biotech, academia. When you have a natural disaster like this, unfortunately, it doesn't matter what market you sit in. So some of the key issues from the case studies really hit on each aspect of the biobanking life cycle, everything from the collection, preservation, transport of the specimens, to how they're stored and how you can ensure their safety long term. The remaining slides that we're going to go over today are really focused on the considerations that one should take into play and take into consideration during study design. So if we look at what makes a biospecimen highly valued, it really has to take multiple perspectives into account. So the legal and ethical issues, the technologies that you're going to use to preserve those specimens or to analyze those specimens, but also the economic factors that impact all of us in making sure that we're getting the most out of every dollar that we spend. And many times as information on the different perspectives for biospecimens are presented, we talk about high-quality specimens, and quality is absolutely essential, but how you define quality will be based on your intended purpose for those specimens. So when we're helping a client design a new collection, we always like to start with the end in mind. Most biobank collections are built off of a primary study that is being performed, and with that primary study, you can plot out the downstream applications that you're going to be performing in order to answer the questions that study is focused on. And then from that downstream applications that are already planned, you can plot what materials you're going to collect, what's the best preservative to use, how best to preserve it. And then from there we can go even further into the weeds of what type of container does that downstream platform require and what are the labeling requirements for that automated instrument to be able to read the information from that vial. But one of the things when you're building a collection for long-term use that you need to consider is that you don't always know, in order to get the most out of your specimens, once your primary questions have been answered, if they've been properly consented and you can now use them to answer new research questions, you don't always know what those downstream applications are going to be. But it is imperative that you still go through the process of what you can envision they could be so that all of the details on the best way to preserve them can be carefully planned. And while we do this, we also need to take into account risk mitigation. There are all different levels of risk mitigation that one can put into place, everything from splitting specimens into multiple aliquots, having those aliquots in different freezers, those freezers in different geographical locations. And during the design phase of the study, you really have to balance what is that risk of loss versus the cost of all of these mitigation steps. So when we look at mitigating against environmental risk, it really is focused on being prepared. In designing a risk mitigation plan, and even designing your initial facility or looking at outsourced partners associated with your specimens, you really have to look at the facility infrastructure and say to yourself, are you in an environment that is at high risk or even moderate risk for a natural disaster? If so, which, and how will you respond to them? And we also have to consider man-made disasters as well, unfortunately, in the environment that we live in today. And so again, taking that into account in choosing the facility that you will build these collections in is critical. And then having predefined action plans for what you will do in different scenarios, whether it's a power outage or a flood, or it could even be a single freezer failure, but making sure that those action plans are well-defined and well-communicated to the team that will be responsible for executing them is key. And in doing this, having written communication plans and strategies, I find, is one of the most absolute critical factors, and that each person knows their roles and responsibilities, because you don't want to be developing a plan while you're in the disaster itself. And prioritizing your activities is another critical factor, so that, again, you can take care of the most important actions first. When you're properly repaired, the response and recovery are already predefined and come very naturally. You can have your response be much more effective and your time to recovery much shorter. The last portion on this slide is testing of your plans. The best laid plans can fail if you don't practice them. And so this is another aspect that we feel very strongly should take place on a regular basis. The first step in building a biological collection is getting an ethical consent from the participant. And some of the challenges that we have is that the therapeutic and diagnostic regulatory requirements associated with the ethical consent from participants is very diverse around the world. We don't yet have harmonization internationally in all of the areas of the world, and so using uniform consents is very difficult to do. But it is important, because in order for a drug or a diagnostic to be approved for use internationally, you often have to have representation from that country's population so that you get sufficient genetic variation across the populations and can obtain a sufficient sample size for your studies. Also, making sure that those approved uses is well annotated for your collection also becomes critical. And some of the key areas that you want to focus on, and these are really hot areas of debate right now in the overall community, and that is the parameters for consent, whether you can use broad consent tactics or whether they need to be very specific and use a dynamic consent model where each individual purpose is individually consented. The return of research results is another arena, because these collections, even in the late-stage clinical trial phases, are still part of research. We do the studies to be able to validate and ensure that the drug or the diagnostic that we want to have approved is accurate. And so is it right to return results that have not been validated to a participant? And we have to keep in mind cultural sensitivity and the politics of other nations. There has been some areas of the world, and still are today, where they don't allow the specimens to leave their country and to help build the biotech technologies within their country. And so each of these things must be taken into consideration. So it's important that when you're looking at a collection to focus on both the consent parameters and your use parameters, particularly if you're using this collection for additional research after the primary questions have been answered. So you need to evaluate, was the original purpose as defined in the informed consent, does it fit what your intended use is today? And what type of regulatory considerations are there in terms of, as you go back to the type of research that was defined? And anonymizing specimens is, of course, one option that some feel can still be done to basically delink specimens from their original private health information. But how are you going to take into account the genetic profile? So if you want to do genetic studies, can it really be anonymous? And you also have to consider, even if you can properly anonymize the specimens, is the data that you're generating from those anonymized specimens with a limited use data set, is it going to bring you sufficient value if you can't rejoin it back to the original data later? So as we look at the considerations for the biospecimens themselves and the integrity of those biospecimens, it really comes back to stability. And just like we test stability for a drug or a reagent or any other type of product, the stability of the analytes you want to measure within that biospecimen should also be tested. And looking at the effects of time and temperature, so that when you're designing your study, you know what pre-analytical factors matter, you know what impact they will have on your downstream analysis. And you need to be able to take both the stability of that biospecimen and logistics into consideration. And when I say logistics, it would be wonderful if you could have every specimen collected and preserved within, 10 minutes, 30 minutes from draw. You pick your time frame. But the bottom line is our clinics are not set up to be able to handle that. And so we have to look at what is logistically feasible across the sites where the specimens will be sourced, and then set the preservation methods, the processing methods, storage configurations in a manner so that we know that the specimens will still be fit for purpose. And documenting this along the way is critical. The International Society of Biological and Environmental Repositories Biospecimen Working Group has actually developed standard nomenclature that can be used for the most common pre-analytical variables. And what you see here for fluid and solid specimens are those that, regardless of what it is you're studying, the disease area of focus, these are always going to be critical pre-analytical variables that should be recorded as you're building your collections. This is in no means meant to be an exhaustive list, but ones that should always be included as a core. And by using SPREC, or the sample pre-analytical code developed by the ISPR working group, it helps ensure that we're all speaking the same language and capturing the same variables, so that as we share specimens with one another to collaborate and accelerate research, we're all speaking that same language. Other considerations during the study design phase is variability. And we're all very familiar with intra and inter-laboratory variability, but making sure that you have a quality assurance plan for your study that takes into account each of the participating sites and has a monitoring plan in place that helps control for that variability and minimize that variability is essential. The one thing many of us need to remember is that the clinical sites where these specimens are being collected and the site-affiliated labs associated with them are working with lots of different active protocols at the same time. And so having indicators that you can track compliance to your protocols and compliance to not only the study protocol, but actually the standard operating procedures that you've determined should be used for your biospecimens should also be taken into consideration. Now, on this slide, we have the typical elements of a standard operating procedure, and everyone knows what goes into the various aspects of these. But the reason we included this in the presentation today is to remind everyone how important it is biospecimens and for the future researchers that will be utilizing those biospecimens that there be sufficient detail so that they can determine if the specimens will be fit for their purpose. To give you an example, we have a collection that we support that is focused on environmental risks associated with cancer. And for that particular study, much of the work had been focused on testing things that could be exposed to various plastics and various materials. However, when a new question was raised about the effects of trace metals and the role it was playing in cancer, the specimens could not be used because they had been collected in cryovials with silicone gaskets, which impacts your ability to be able to accurately measure those trace metals. So making sure that you're planning for these types of things ahead and that you have that kind of detail within your procedures becomes important so that you know whether or not the specimens will be suitable. Also, your labeling. Again, this is something that is incredibly basic, but also incredibly important. So having unique identifiers associated with each specimen allows you to ensure you can maintain a chain of custody for that particular specimen and can distinguish it from its sister aliquots that were created when the specimen was initially preserved. Having that identifier be a coded identifier is very common practice today. I think we're all very well aware that using de-identified specimens should be done. What we do still see, however, is people building logic into that code. And when you build logic into the code, it makes it much easier to be able to decode information, even if it's just geographic area as an example, or gender. But again, that could introduce bias into interpretation of results. And making sure that as you're assigning identifiers, you're actually leaving room for later quality control specimens to be inserted can be critical. And of course, that label has to be readable by all parties and instruments for which you expect that specimen to be exposed to. And so, again, making sure that everybody in the chain can read it. For data collection and data management plans, we're all very focused on making sure that we get the right data elements collected, but making sure that there are clear data dictionaries that define each of the fields for the type of information that you're capturing is important, as well as the technology that will be used, the roles and responsibilities of who will capture the information, and how you'll properly quality control and monitor the integrity of that data needs to be taken into consideration. And making sure that it links to your biospecimen collections. And I say it that way because it can often be different parties that are holding the data repository from the physical repository. And so making sure that the data attributes that are associated with the particular patient or that patient sample and the attributes of that specimen itself, you need to make sure that those two parties can continuously link them. some additional considerations for your quality assurance plan. Making sure that you have data to support your chosen method, so pilot studies will be able to demonstrate that you have controlled for variability, having your training and competency plans incorporated, as well as tracking and trending of your key quality indicators. These key quality indicators, the most common we see, some are performance indicators such as accrual against targets, sample yields, but also results of your quality control and monitoring practices become other important indicators. And I don't expect anyone to be able to read the specifics on this slide, but this is more just to illustrate a very simple exercise that one can do to plot out the various types of specimens that are required for a primary study, and then being able to look at the impact factors of the pre-analytical variables to ensure that, number one, you're collecting a sufficient amount of specimens, and number two, you have the right processing methods and quality control checks in place to know if that specimen is not going to be appropriate for analysis. And then also, one of the things is we started this presentation, or I started this portion of this presentation, defining a highly valued specimen. And one of the things about value of these specimens is that we get a lot more of it if we're able to use them for additional purposes in the future. Even if it's discovery-type research, of being able to scan these libraries of specimens can be a very powerful resource that allows you to accelerate your development life cycle. And so when you look at utilizing your existing collections, we often work a lot with clients to evaluate what clinical data they have available, whether or not the specimens are consented for that broader use that it could now be used for, and whether or not the specimens themselves would be suitable. And these types of analysis or utilization assessments, we feel very strongly should be performed periodically, because there's no reason to hang on to a specimen that does not have clinical data available or was not consented for broader use if you don't think that there's a relevant science or a future scientific question that fits within that use for which they could be used. And so that becomes how we get more value out of our existing resources. And Pia, I'll turn it back over to you.
Thanks, Kathi. Kathi, you did a great job in sort of raising our awareness as it relates to both sample and storage consideration, highlighting that fit for purpose and what I extend on to that, which is fit for the future, right? So I hope that collectively Precision has helped the folks on the line raise awareness, create more alignment between these two worlds of biobanking and companion diagnostic development. Hopefully either close some gaps or open some conversations that need to occur in your organization and begin to think about the quality and the value, but nonetheless maintaining that sort of high-valued biospecimen collection is critical to not only the conversation but the successful development of companion diagnostics. So thanks again. We're going to move to the question and answer section.
So, Kathi and Pia, how much of the research will have a mandatory companion diagnostic in the next 5 to 10 years?
That's a great question, and I wish I had a great answer for you. Say that in the next 5 to 10 years, it will be mandatory that 99% of drug developers will have to attach a companion diagnostic. What I can point to is that currently 1% of the drugs on the market today have a companion attached to them. So don't despair. What I'll point to is that 80% of drug developers are developing their compounds with a biomarker. And a biomarker can serve as a platform for the future use of a companion diagnostic program. So that, to me, is moving in the right direction. We're seeing utilization of a biomarker strategy, formalization of a companion diagnostic, in this environment, and I think there will be exponential growth in this field. Thanks for the question.
Great. And moving on to the next one. What are the most effective approaches currently available to improve the quality of human tissue specimens and pertinent clinical pathological data from patients enrolled in clinical trials?
Well, Teresa, I can take this one. I think one of the most effective tools, the most effective approaches that one can have is creating an effective quality assurance plan. And this quality assurance plan should encompass all phases of the specimen life cycle. And I really believe in using a risk management approach to identify the areas of potential risk during the design phase of the study. And doing this and having a detailed quality assurance plan will allow you to mitigate against those risks and increase your chances of success. You know, we talked about the Hurricane Sandy and the impact that had in New York. And as I mentioned, they had thought, well, gosh, with our generator up on the roof, but the fuel pumps down on the ground, what would we do if we encountered both of those situations of an extended power outage and flooding? I'm sure they would have designed things differently.
Great. Thank you, Kathi. Could either of you give a brief impact on how pharmacogenomics will affect this area?
Sure, I'll take that, Kathi, thanks. So regardless, I'll take a step back and say, you know, the core thinking on companion diagnostics centers around three areas, right? It is the assay or the content, right, of the test. Secondarily, I don't see the world of the content getting any less complex. The existing companions on the market today focus on one drug, one mutation. It can be an IHC test, a fish-based test, a PCR test, and therein goes more of the more simplistic technologies. But rather the platform as well. I don't see the platform getting less complicated. And so the question is understanding the scope and the emphasis of pharmacogenomics as it relates to biospecimens. So I would sort of point to ensuring that we're storing and preserving these, because the variables at play on the pharmacogenomic side are only going to get more complicated. So if we didn't take the steps that Kathi elucidated on the pre-analytical variables to ensure that we could utilize these wonderful sort of far-reaching technologies, so again, I'm trying to bring that into that world so that pharmacogenomic piece as it stands today is not going to get any simpler. The technology is going to get more complex, and so preparing the biobank and the specimens inside of there is critically important to the uptake and access of that.
Great. Thank you. The next question is, what is the future of biobanks supplying greater pre-analytical electronic data?
And Teresa, I mentioned earlier in the presentation that the SPRUT code that was developed by the ISPR Biospecimen Working Group, and ISPR has started the movement to standardize nomenclature for some of those most common pre-analytical variables. But as I mentioned, they're by no means all of the information that we need to capture associated with a specimen. Which pre-analytical variables are going to be important can be impacted quite a bit by the type of things that you're going to measure. So to give you an example, is the position of a donor during a blood collection. If you were going to be measuring infectious disease analyte associated with that participant from their blood, it's not going to matter which position they were in during the collection process. But if you want to look at endocrine levels, that could very much be impacted. And so capturing information on that collection process would allow you to determine whether or not that was a factor for your analysis.
Okay, great. Another question that has just come up is, if a patient sample is used in the study and later that particular patient retrieves his or her consent, Do we have to go back and take out the sample from all studies and reanalyze the data, or is it that it cannot be used anymore for the studies from the time the consent was retrieved?
So this is an excellent question, and one of those areas that, again, I think has had a lot of debate, because this circumstance 10 years ago was not typically included in the informed consent, and today it is. And so what we often see is that built into the informed consent, there actually must be a requirement now of where you inform the participant what if they want to withdraw from the study, how will everything be able to be withdrawn from the study. And the most common language that we see is that if you want to withdraw consent, any specimens that have not already been consumed will be discarded, but it may not be possible to discard the data. So again, that should just be very forthright within the consent and then clearly documented as part of your annotation associated with that specimen, your consent annotation.
Finally, I think we have one more question before wrapping up. So I'll pose to Pia and Kathi, what do you predict the future will be like in the biobanking arena?
I really see biobanking, I think the world sees biobanking these days as a rapidly evolving field. Over the last decade, there's been a huge increase in the awareness of the importance that biospecimens play in medical research. And over the next decade, I personally think that we're going to see the development of standards and regulations similar to those governed by the FDA or that fall under CLIA for testing laboratories. We need to remember that a biospecimen is a representation of the patient, and our focus really needs to be on ensuring that we're obtaining an accurate result when we're utilizing their samples for research, rather than measuring the effects of the pre-analytical variables that we could have introduced during the collection and preservation process.
Great. It looks like we actually have a little bit more time, so I'll ask one that's just popped up on our screen. Is there a program to define the data standards for biospecimen, specifically helpful when you collaborate or procure biospecimens externally, similar to CDISC or HL7 for clinical trials and healthcare data?
So I've mentioned the SPREC, which is a standard nomenclature for some of those pre-analytical variables. There is also a group or a set of data standards. STARD is the acronym, where they have defined parameters. The College of American Pathologists, through one of their focus groups as well, has also recently developed a very detailed listing of the data fields that should be collected, both from a demographic standpoint. It actually delves into some clinical data as well, and then, of course, very detailed information about the biospecimens and the processes. That publication is getting ready to come out, but has not yet been published. But we could certainly provide that information once it has. We could provide a link to that on our website.
Great. Well, with that, I'll thank Pia and Kathi for hosting this webinar, as well as CHI for putting this on for us here at Precision for Medicine. It's a very dynamic industry, and we look forward to hearing what happens in the future. So we'll take all the questions that we received from the chat panel, as well as those that came in when you registered, and provide follow-up answers to those, as well as you can expect to receive an email with a copy of the recorded webinar and any follow-up items that Pia and Kathi may feel is relevant as you review. Thank you for joining, and have a great rest of the day.